So let's go ahead and generalize this a little bit. So we have two-factor ANOVA, and let's look at all the possible outcomes we could have in terms of significance. So in this case, our factor A we'll call food, and on our factor B we'll call the feeding frequency, 1 or 2 times per day, and then factor AB is the interaction. So we really have 5 possibilities here. Factor A, the food, could be not significant. Factor B, the feeding, could be significant, and the interaction could not be significant. And again, technically we would look right to left, so that's actually what I'll do from here on out. So go to the second row. The interaction could not be significant, the factor B, the feeding, could not be significant, and factor A could not be significant. That's another option. Go down to the third row. Again, the interaction would not be significant, factor B would not be significant, and maybe in this case factor A is significant. Go down to the fourth row. The interaction is not significant, but factor B and A are both significant. And then the final possibility is where the interaction is significant. And notice, once the interaction is significant, we don't even look, we don't even consider the two factors individually. If the interaction is significant, the individual factors cannot be analyzed because they are too intertwined. They are too confounded together. We cannot look at them separately. So how would this actually look on an interaction graph? Let's really understand how these are made. So you can see here along the bottom, we have our column effects. Remember, our column was our plant food. So in this case, I labeled them A, B, and C, same thing. So food A, food B, and food C, those are our column effects. Each line is our row effects. So remember, our rows in our table were the frequency with which we fed the plants. So we have column effects along the bottom, and the row effects are our actual lines. Now in the axis over here on the left, you can see that that is our growth in centimeters. And we're going to be plotting the cell means on this graph. Now, when we go to our column effects, what we're going to look for is, are the lines increasing, going up, or decreasing, coming down? Or are they just flat, like you see them here? Now for the row effects, what we're going to look for is, are they far apart, or are they close together? So think about this for a minute. Look at the column effects up and down arrow. If the plant grows more as we go from food A to food B to food C, we would expect those lines to go up and to the right. Well, if the plants get taller as we go from food A to food B to food C, we would probably think that the plant food makes a difference. It does have an effect. Now if it goes down and to the right or something like that, then again we would say it probably has an effect, in this case the opposite, that the plant growth actually goes down as we go from plant food A to B to C. Now of course it can be sort of up and down too, it doesn't have to be a straight line either way. Now what about the row effects? If the row effects are far apart, it would seem that the frequency with which we feed the plants makes a difference. So in this case, it seems like the 2 feeding line is much higher than the 1 feeding line. So it would appear, based on this graph, that the frequency with which we feed our plants does make a difference. And again, these are just graphical models so we can understand how they're sort of made. So let's look at this one. And this is some hypothetical data just to sort of get across how these graphs are made. So for 1 feeding, we had the growth of 50 centimeters for A, 50 centimeters for B, and 50 centimeters for C. So you can see these points right here. Now for 2 feedings, we had 70 centimeters for food A, 70 for food B, and 70 for food C. That's the top line. Now, do these lines go up and to the right or down and to the right? Are they not flat, basically? Well, the lines are flat. So it seems that there's no change across the different plant foods. So those are our columns, C. So there's no change across plant foods because the lines are flat. Now what about the distance between the lines? Well, these are our row effects. The lines are far apart. They change across feedings, so our rows. And that should make sense based on our data up there. We can see that the 2 feeding row is consistently 20 centimeters higher than the 1 feeding row. So what we conclude based on a graph that looks something like this? Well, the lines do not cross, we know that. So we would say that our columns are not significant probably. We wouldn't know until we did the actual analysis, but it appears our columns, or our plant foods, are not significant because the growth does not change at all across the plant foods. What about our row factor? Well, in this case, it does appear the row factor is significant. The lines are far apart. The 2 feeding line is pretty far above the 1 feeding line. And then we already said that the lines do not cross or are not going to cross, so there is no interaction here. There is no change between the terms as we go across the graph. No interaction. So what about one that looks like this? So here's our hypothetical data. So for 1 feeding per day, we had 60, 60, 60. And then for 2 feedings per day, we had 62, 62, 62. That's the top line. Are the lines increasing? No. Are the lines decreasing? No. So what does that tell us? Well, the lines are flat, so that means that the growth of the plants, again, does not change as we go from plant food to plant food. It's always 60, 60, 60 or 62, 62, 62. There's no change there. Now what about the distance between the lines? Again, this is our feeding lines. Well, the lines are very close together. There's really no significant change across the feedings. 62 is not that much different than 60. So in this case, of course, the lines do not cross. And think about this in practical terms. Does it matter which plant food we use? No. Does it really matter which feeding we use necessarily? 1 or 2? Not really. We're getting about the same growth from 1 feeding as 2. 1 feeding is cheaper, so we'll probably just feed the plants once because it's not that big a deal to get 2 more centimeters and a whole nother feeding. And of course, there's no interaction. So how do we interpret this? Well, the columns, the foods, that's not significant. The lines are flat. The row factor, the frequency at which we feed the plants, that's not significant. And of course, there is no interaction. So neither main effect or neither of the interaction is not significant. And that's it. What about one that looked like this? So here's our data. So for 1 feeding per day, it was 50, then 55, and then 60. And then for 2 feedings per day, 52, 57, 62. Are our lines increasing up and to the right? Well, yes, they are. Are they down to the right? No. But they are increasing up and to the right. So what does this seem to indicate? Well, the lines rise. So in this case, there is some change across the plant foods. So plant food A seems to be the lowest in growth, plant food B seems to be the next highest in growth, and plant food C seems to be the highest in growth. There is change across the columns as we go from A to B to C. Now what about the distance between the lines? Well, it's not very much. It's still 2 again. So the lines are really close together, so it does not seem that the number of times we feed the plants per day makes that much of a difference. Again, just 2 centimeters. And of course, the lines do not cross. So 2 feedings per day is consistently better than 1 feeding per day, even though it's not by that much. So we could probably conclude here that the columns, C, the plant foods, are significant. So the growth does go up as we go from food A to food B to food C. Now the rows, or the number of feedings per day, is not significant because the lines are very close together. And of course there is no interaction because the 2 feeding line is always above the 1 feeding line. So in this case, all else being equal, we would probably select food C and feed the plants once per day, because it was consistently the highest, but feeding it twice doesn't really get us anything. So how about this one? So here's our data. So for one feeding we have 50, 55, and 60. That's the line on the bottom. And then for 2 feedings per day we have 70 centimeters, 75, and 80. So are the lines going up and to the right or down and to the left? Well, yeah, they're going up and to the right again. So what can we conclude from that? Well, the lines rise. So there is a change in growth, again, across the plant foods. As we go from A to B to C, both the 1 feeding line and the 2 feeding line go up and to the right. What about the distance between the lines? So our feeding lines. Well, they're pretty far apart. So it does seem that 2 feedings per day generates a lot more growth. And do the lines cross? No. Again, because 2 feedings per day is consistently higher than the 1 feeding per day line as we go across. So what do we conclude here? Well, the column effects, the foods, are significant. That's because the lines go up and to the right. Now the row effect, or the feeding frequency, is also significant because the lines are far apart. The 2 feeding line is always above and pretty much higher, 20 centimeters higher, than the 1 feeding line. Now there is no interaction here because the 2 feeding line is always above the 1 feeding line. They do not cross. Now, how about this one? This one looks different. So for the 1 feeding per day, we had growth of 50 for plant food A, 60 for plant food B, and 70 for plant food C. Now for the 2 feeding line, we had 70 for plant food A, 60 for plant food B, and 50 centimeters for plant food C. Well, that's kind of weird, isn't it? So, do the lines cross? Yes. And notice, we stop there. Because we really couldn't say that there's an upward or downward pattern overall, could we? No. We really couldn't gauge if the lines are far apart because, well, they cross. So we look at the interaction first, the lines cross, and we stop. So our interaction is significant. Neither line is above the other one consistently across the graph. They cross, so therefore we would anticipate a significant interaction. And if the interaction is significant, we stop there. We do not analyze the columns or the rows. Let's look at a quick example, just another one, so you can see how this works. We won't go through all that again. But here we have a table, and these are shoe stores. And on the top in the columns, we have the number of competitors, of competitor shoe stores, within a 5-mile radius of each location. So 0 competitors in a 5-mile radius, 1 competitor, 2 or 3 or more. On the left-hand side, our rows, we have the type of store it is. So we have a standalone store, which is just a store by itself that's sitting out somewhere. Then we have a store that's in the mall, so maybe in a suburban mall or something like that. And then finally we have stores that are located in a downtown sort of urban area. So those are our 2 factors, the number of competitors in a 5-mile radius and the type of store it is. And then here we have the average shoe sales per week. So we can see that standalone store with 0 competitors around it had an average shoe pair sale of 38.667 pairs of shoes, and so on and so forth. So we can see that each column has its own mean. So the 0 competitor column has 30.444, 1 competitor column has 29.556 pairs of shoes on average, etc., and so on. Also, the type of store has its own mean here in the end. And then down the bottom right, we have the overall mean for all the shoe stores. And there are 10 shoe stores in each cell. So what you're looking at here are cell means. Cell means. So 10 stores are combined to generate these means in this table. So let's go ahead and look at our estimated marginal means graph that comes out of SPSS. Now I've tried to color code these so you can see exactly where everything is. So let's go ahead and look at the standalone row. That's the blue line. It's also the blue row in the table over here on the left. So you can see we have 38.667, 36, 52.667, and 42. So if we look at the blue line over here on the right, that follows that pattern. 38, 36, 53 about, and then 42. And of course the green row is the mall. So in the table we have 26, 31, 47, 46. And if we look at the row over here on the right in the graph, the green line follows those numbers exactly. Then we have the downtown line. So 26, 21, 27, 27. So this sort of light brown line along the bottom in our marginal means follows those numbers. Now, let's go ahead and look at this in a different way. So here is the same thing we had before. And I want you to follow along. This is really important to understanding what we're trying to learn here. So let's take the overall mean. So the overall mean is 35.278. And I gave that a dashed line. Now let's go ahead and take that dashed line and put it on our marginal means graph. So we'll put it right there. So that is a value of 35.278 approximately, and we'll put that right across our marginal means graph, and again that's the overall mean for all stores. Now let's go ahead and give the 0 column a red dot. So the 0 competitors column had a mean of 30.444. Now let's go ahead and represent that on our graph over here on the right. So boop, there it is. Now, if you look at our column, if you put your hand over everything to the right of 0, forget it exists. Now we go ahead and put a red dot where our column mean is for all those 0 points. So you can see that we have 26 for the green, we have 26.6-ish for the light brown, and then 38.667 for the blue. The red dot there is the mean for that column. Let's go ahead and do the same thing for one competitor. And there it is. So 29.556. So that is the mean of those 3 dots that make up the one competitor column. Now for 2 competitors, we'll put a green dot. And there it is. So that is the mean for those 3 points in the 2 column. And then a yellow dot for 3 or more. So put that right there. So those 4 dots are the column means for our number of stores. So 30.44, 29.556, 42.556, and 38.556. So red, blue, green, and yellow. Now, let's do the same for our rows. So we'll give the standalone stores a light blue star. So let's go ahead and put that mean there. So what is that? Well, that is the mean of the blue line. So the 4 points that make up the blue line, so it starts at around 40, goes down, then goes way up, and then comes down again. The mean of those 4 points is 42.333, and that's about right there relative to the values on our graph. Let's do the same thing for the mall. So we can see that the mean for the mall is 37.667, and we'll put that right there. So that green star is the mean of the 4 points that make up the green line. And finally, the downtown. We'll give that sort of a pink color. So we'll do that, and that is the mean of those 4 points. So the 4 points that make up the downtown line there along the bottom, that mean is there. So really, take a look at that. That's really important for understanding how this works. All right, so go ahead and put everything back so you can see how it all fills in. And there we go. Now, on the lower left, we have the output from SPSS. So what's the first thing we look at? The interaction term, exactly. So we look at competitors by location. There's our interaction term highlighted in yellow. So we go across. Its significance is 0.016 with an F of 3.315. So if we're at a 0.05 significance level, that interaction term is significant. So again, we would not go on and evaluate the two individual factors independently. So we'll say the interaction is significant, and we stop there. Now there are ways to put the sort of effect size into your research paper or something like that for those other terms, but we're not going to go into that. But really in the analysis, we look at the interaction first. Now let's go back to our food data, our plant food. So here's everything we had from the plant food. Same idea. So we're going to put our overall mean there where the black dotted line is. So our overall mean is 59.09. So we'll put it right across there. Now for the awesome advantage column, a red dot again, that column had a mean of 63.5. So we'll put that right there. So again, that's the mean of those 2 points that make up the AA column. For BB, for big buds, we had a column mean of 64.75. So we'll go ahead and put that there. So again, that's the mean of those 2 cell means. And then for food CC, same idea. It is there. So again, those are our column means. So those are the means of the 2 points that make up each column. Now let's do the rows. So for the 1 feeding row, we're going to put that there. So it had a mean of 60, so it's almost right on top of our overall mean of 59.09. And then our 2 feeding row had a mean of 58.17. So it's almost right on top of that one too. Now let's go ahead and look at our SPSS output. So again, we look at the interaction first. So there it is. So feedings by plant food. And we see that we have an F value of 9.333. And again, our significance is 0.000, so that is obviously significant. So we would really stop there in our analysis. We would not go on to evaluate the individual effects. But for this problem, we're going to take a look at that, because this is really important to understanding how all this fits together. Look over at our graph here on the right. Okay? Now look at our overall mean line, the black dashed line. Which of the 2 are on average further away from that? The column means, so the types of plant food, or the row means, the frequency at which we feed? Well, according to my vision, it looks like there is more variability in the type of plant food. So you can see that the distance from the dashed line to the dots is much further than the dashed line to the stars. Now, go over and look at our SPSS output again. Look at the significance level for feedings. Those are our stars. So we have an F of 0.578 and a significance value of 0.451. Those are our stars. Now I want you to connect the F and significance for the feedings on the left to where the stars are relative to the dashed line over here on the right. Are they far away? No. Is there a lot of variability in them? No. They're almost right on top of the overall mean. That's why it's not significant, because they're right on top of the overall mean. There is no variability there. There's no difference there. Now, look at the plant food factor. It has an F of 16.526 and a significance value of 0.000. So if we were looking at it on its own, we would definitely say it's significant. But of course, there's an interaction, so we don't do that. But look at how the SPSS output corresponds to the means over here on the right. So for the plant foods, there's a lot of variability among them. The red and the blue are above the overall mean. The green is significantly below the overall mean. There's a lot of distance there. There's a lot of variability there. there's a lot of change there as it corresponds to the overall mean, as compared to the stars. And that's how we can interpret visually the SPSS output over here on the left. If you can understand what's going on in this graph, in this slide here, you've got it. Alright, so let's sum this up and get done with this video. So, interaction graphs are an easy way to eyeball the relationships between cell means, column means, row means, and the overall mean. Now, crossed row lines indicate usually an interaction. Column means spread far apart from the overall mean, like we saw last time, indicate a significant column factor. Row means spread far apart from the overall mean usually indicate a significant row factor. So remember that interaction graph with the dots and the stars. The further away the dots and the stars are from that overall mean line, the more likely those are to be significant. And of course, it depends on the data as far as whether or not there is an interaction. Because both main effects can be significant without there being a significant interaction. It just depends on where those dots and stars are relative to each other. That's the whole point. So just because each effect is significant does not mean there's an interaction that's significant. That's important to remember. Now, if an interaction exists and it is significant, the row and column effects cannot be evaluated individually. The main effects are too intertwined. They are too confounded together to look at individually because the values change as you go across, and you really can't untangle it. It's like a knot you cannot untangle. Okay, so that wraps up our video on the analysis of variance, part 2, where we're looking at graphs of marginal means. And again, I hope that you realize that this sort of visual tool can really key you in as to what's going on in your data. So if you make a simple graph of marginal means, it can tell you a lot about your data before you even run it through a statistical software package. Now, I will say that the examples we used here were simple. We didn't have more than 3 factors in any variable, and they were pretty obvious. Now, you can have multiple levels in a variable. So you could have, 6 competitor levels and 5 different types of stores if you wanted to. And the interaction graph may be hard to decipher if you have a lot of levels in your variables. So the ones we were looking at are relatively simple. But once you pick up the pattern, even the more complex ones, you'll be able to see these patterns in the interaction graphs, or the graphs of marginal means, that again will help you interpret what's going on with your data. So, thank you very much for watching. I wish you the best of luck in your work and in your studies, and I look forward to seeing you again next time.